Hi, my name is Doreen, and today I'll be presenting on MRSA. MRSA is methicillin-resistant Staphylococcus aureus. This is not just a textbook bacteria, it's one of the most dangerous drug-resistant pathogens in the world. To put it in perspective, MRSA causes roughly about 90,000 infections and 19,000 deaths in the US every year. It exists in two main forms. There is hospital-acquired and community-acquired, which I'll break down shortly. Now, S. aureus belongs to the family Staphylococci. It's a gram-positive cocci, meaning it stains purple and clusters in grape-like arrangements. It's a facultative anaerobic, so it can survive with or without oxygen, and it's non-motile. What makes MRSA specifically dangerous is its arsenal or virulence factors. That is, protein A helps it hide from our immune system, and coagulase lets it clot plasma to create a protective barrier. And alpha hemolysis destroys the red blood cells. Most critically, the mecA gene encodes a modified protein called PPB2A, which is a molecular key to its antibiotic resistance. Now, MRSA is an opportunistic pathogen that can infect virtually any part of the body. The most common presentation is on the skin and soft tissue infections. Think about boils and abscess with an incubation period of 1 to 10 days, but it can escalate quickly. Once it enters the bloodstream, it can cause bacterium and sepsis, with a mortality rate as high as 50%. It can infect heart valves, causing endocarditis, the lungs, causing necrotizing pneumonia, and in severe toxic producing cases, it triggers toxic shock syndromes. This range of disease is what makes it so clinically challenging. Now, epidemiology and the global burden. There are two distinct epidemiological patterns. That is, hospital-acquired MRSA tends to strike patients who are already vulnerable. That is, people on ventilators, post-surgical patients, and ICU patients. And it accounts to about 31% of all S. aureus bloodstream infections globally. Then the community-acquired MRSA, on the other hand, affects otherwise healthy people. That is, athletes, military personnel, children. So the USA 300 strain is the dominant community strain in the United States. Globally, antimicrobial resistance, of which MRSA is a major contributor, it kills approximately 700,000 people per year, a number which is projected to rise dramatically if resistance continues to be unchecked. On the next slide, we want to talk about the source and transmission, where MRSA is a carrier-type pathogen, which means it lives in humans. About 30% of the general population asymptomatically carries S. aureus in their nasal passages, and roughly 1 to 3% carries MRSA specifically. Transmission happens primarily through direct skin-to-skin contact with an infected wound, or by touching contaminated surfaces and shared items like towels or athletic equipment. Healthcare workers can inadvertently carry and spread in between patients. Prevention is straightforward in principle. That is, hand hygiene is the most single effective intervention. In healthcare settings, contact precautions and active surveillance of incoming patients are standard. For high-risk surgical patients, decolonization with intranasal and chlorhexidine bathing is used proactively. So people need to sanitize every so often, every time they are in contact. So, about the laboratory diagnosis. There are four main methods used to diagnose MRSA. First, there is the mannitol salt alga, which is a selective medium with high salt that only Staphylococcus can survive on. As a rose ferments the mannitol and turns the agar yellow. So this test, it will make the bacteria look to be yellow, giving a presumptive ID. The second diagnosis is through the Kirby-Bauer disk diffusion, and then there is also the PCR method of diagnosing it, and then the MRSA chrome agar method of diagnosis, where the colonies will appear pink in color. So this last test, the MRSA chrome agar, is very fast, visual, and highly specific. Now we go to the treatment and the therapy for MRSA. Because MRSA is resistant to all standard penicillins and cephalosporins, treatment requires entirely different drug classes. Vancomycin is the first-line IV treatment for serious infections. It's a glycopeptide that blocks cell wall synthesis by binding to a different target than the B-lactams. And then for community-acquired skin infections, the oral TMPs, or we call them the clindamycin, are often first choices. Cetra, cet, Ceftrolin is a newer fifth generation that is unique among the B-lactams, usually binding to the PBP2A and works against the MRSA. It's important to note that there is no licensed vaccine for MRSA, and surviving an infection does not confer lasting protective immunity, which is part of why it remains so hard to control. Now, what is the emerging research and new data on this disease? For new data, I would like to highlight a 2023 study by Ravera and colleagues published in the journal Molecules. They demonstrated that the two plant-derived flavonoids, that is quercetin and cicerol, can inhibit the FOF1 ATP synthesis enzymes in the MRSA. By blocking ATPA production, they essentially starve the bacteria of energy. This is exciting because it represents a completely new mechanism of action during drug-resistant strain. Other active research areas include the phage therapy, where engineered viruses are used to infect and kill MRSA. Why is MRSA so dangerous? To understand why MRSA is so hard to treat, you need to understand the PBP2A. Normal bacteria will use penicillin binding proteins to build their cell walls. The B-lactam antibiotics like methicillin, oxalin, and all penicillins work by binding to and disabling those proteins. But MRSA acquired through a mobile genetic element called the SEC mecA. These genes encode the PBP, which is structurally altered version of the protein that simply cannot bind. So the bacteria keeps building their cell walls and dividing completely unaffected. On top of that, MRSA strains are frequently co-resistant to aminoglycosides, making treatment options progressively more limited. The FDA has classified MRSA as a serious threat, and the CDC calls it an urgent public health priority. So, in conclusion, I will say that MRSA is a gram-positive, is facultatively anaerobic cocci that has evolved extraordinary resistance through the mecA gene and the PBP2A. It causes diseases ranging from skin infections to sepsis, and it kills nearly 19,000 Americans every year. It spreads through direct contact, and colonizes healthy carriers asymptomatically. We diagnose it with culture, the Kirby-Bauer testing, and PCR. We treat it with vancomycin and other non-β-lactam agents with no vaccines currently available. And emerging research like the ATP synthase inhibition study offers real hope for new treatment strategies. MRSA is a reminder that as we develop antibiotics, bacteria evolve, and staying ahead of resistance requires constant scientific vigilance. Thank you.